Okay, welcome back everybody to this lecture series on biotechnology and its applications. As I mentioned earlier, my name is Abhishek. I'm a fourth year BSMS biology major at IISER Trivandrum. And in the last lecture, which was incidentally the first, we looked at the basics or the fundamental concepts of genetic engineering and recombinant DNA technology. And I tried to give you a flavor of what is to be expected in this topic as we go forward. And I left you with these three interesting points. That is the basic steps in genetically modifying an organism, which basically means changing the genetic composition of that organism by introducing or removing specific DNA sequences or parts of its genome. I said, with the help of the example of, let's say, insulin for treating people with diabetes. One way is to, of course, extract insulin from other organisms like pigs. But then if you introduce that insulin into a human, you can have adverse immunologic reactions. Also, you cannot really extract insulin in very large quantities with these methods. So one can think of, it's actually rather ingenious, one can think of alternate methods where you identify DNA with desirable genes. In this case, you could take the human insulin gene, and let's say by some incredible engineering, you could introduce this gene into a bacteria which rapidly divides. And let's say even more magically, this bacteria could actually take this gene and use it to produce protein. And since the bacteria keeps dividing and it keeps expressing this gene, you could create a perpetual cycle where you could continuously produce insulin protein from human insulin gene. Then if you could somehow purify this protein, then you could use it to treat people with insulin without any adverse immunologic effects. Because of course, the gene comes from humans, so it's a human insulin gene. So you're not going to have any kind of immune response as you did when you extracted insulin from pigs. So this leads to this kind of workflow of sorts, which is first starts with identifying DNA with desirable genes, introducing this DNA into a host, which in this case would be the bacteria which you want to divide and use this insulin gene to produce insulin. Then, of course, for this to happen, the DNA has to be maintained in the host and the DNA has to be transferred from one generation to the next. Because if the bacteria divides and the DNA does not go from the parent to the daughter cells, there's no point. An additional 2 points, as I mentioned just now, could be that the host should be able to use this DNA to produce your protein of interest, and then you should be able to purify the protein. So here is where the intro kind of ends of sorts, and we start moving into the more nitty-gritty of how exactly this kind of genetic engineering or biotechnology, recombinant DNA technology, whatever fancy word you want to use, how exactly is this possible? It's time to get out my pen. So the first tool, we're now going to be talking about a series of tools when it comes to recombinant DNA technology, with the first one being restriction enzymes. So the first thing you should know about restriction enzymes is that they are proteins, and proteins are integral and vital to all cellular functions. So proteins carry out very diverse roles in the cell. Whichever cellular process you're studying, you're definitely coming across multiple different proteins. This is one particular class of protein called restriction enzymes. And in layman terms, they would be called molecular scissors. So it's very easy to understand. Scissors are used to cut things. So if these proteins are called molecular scissors, they must cut something. So what do they cut? In essence, these molecular scissors can cut the DNA at specific and well-defined sites. So your immediate question would be, okay, so what do we mean by well-defined sites? Okay, so before that, what do we mean by a site on the DNA? A site is nothing but any location on the DNA. DNA is a long stretch. So basically, you have a long stretch of DNA, which is just some sequence of the bases A, B, C, and G. And a site is any particular location on the DNA. So if let's say I take a site of 6 base pairs on a large DNA stretch, I take a site of 6 base pairs, then that is going to have some random combination of A, T, C, and G in some order, maybe AAA, TTT, or A, T, C, G, T, A. And if I take another site somewhere else on the DNA, let's say to the left or to the right, I take another site, what is the probability that it's going to have exactly the same sequence as this site in the middle? It's very low. You can work it out yourself. There are only 4 possibilities, and the probability of getting the exact same sequence when you take 2 random sites of the same length, I should say, is rather low. So these restriction enzymes do not cut DNA at any particular site. It's not like whichever location on the DNA you give them, they can cut. No, they cut the DNA at specific well-defined sites. So we know what a site is now. It's any location on the DNA. And I've told you that restriction enzymes are essentially molecular scissors which can cut the DNA at well-defined sites. So well-defined locations. Now you must ask, so what defines these locations on the DNA? It's very simple. If you think about it, what could possibly define a location on the DNA? The obvious answer is it must be its sequence. The sequence is what can define location on the DNA. For example, I can say go to this piece of DNA and find a site of length 6 with this particular sequence AAA, TTT. Find all such sites because, of course, probability dictates that there will be more than one such site. So I shouldn't phrase it like that. As you increase the length of the overall DNA sequence, the probability that you find a particular site of a given length increases simply because the number of bases increases. So if I tell you, it should be obvious that sites of shorter lengths occur more frequently than sites of longer lengths. So if I ask you to find every site of sequence AA on the DNA, it's much more likely that you'll find many more of those than, let's say, a site AATTGCTAC, simply because, well, you can work it out based on probability. It's rather simple. So when I say these restriction enzymes cut DNA at specific and well-defined sites, a site is nothing but a location on the DNA, and we're defining that site by, I made a mistake earlier, by two things. Number one, its length, and number two, its sequence. So this is what defines a particular site. So there's a lot of text here. Apologies for that. We'll get to that in a minute. But before that, I'd like to explain to you in more detail exactly what I meant. So this is taken from the NCERT textbook. I thought that would be of some use to you to have figures from your textbook so that you feel more comfortable when you look at them in the future. Now, at the present moment, forget about this vector DNA and foreign DNA and all that. We'll come to those in the future. For now, just focus here and here. Okay, so we are talking about restriction enzymes. The restriction enzyme in question right now is EcoR1. As I mentioned before, each restriction enzyme can cut the DNA at a well-defined site. The site is defined by its length and DNA sequence. Now, okay, just a fact to remember here, the site that the restriction enzyme can cut is called its recognition site or recognition sequence. Every restriction enzyme has a unique recognition sequence. All right. So what I'm saying is, if you have a restriction enzyme with a particular recognition sequence and you have a long stretch of DNA, and this particular recognition sequence occurs, let's say, 3 times in this larger stretch, then your restriction enzyme will cut the DNA 3 times, because every time it encounters that particular site of a given length and sequence, your restriction enzyme will cut the DNA at that site. So now an example, the restriction or the recognition sequence for EcoR1 is GAATTC-CTT-AAG. So essentially, if you have EcoR1 under the right conditions for it to function, because you must remember enzymes function at very specific temperature and pH conditions. So if you provide the optimal, or let's say you provide good enough conditions for EcoR1 to function, and you have a DNA sequence, or you have a piece of DNA, wherever you have the sequence GAATTC-CTT-AAG, EcoR1 is going to cut it in a very, very specific manner. So I can revise the definition a little bit. So these cut the DNA at specific, well-defined sites in a specific manner. So you will always have EcoR1 cut between G and A and GNA. Here as well, GNA, there's a small mistake in the textbook. It should be G and A. So once these cuts are made, you can easily appreciate that this piece will now float away, and you will get this formation GCTTAA on the vector. Again, forget about what vector and foreign DNA mean for now, those will become clearer. And on the foreign DNA, you'll get GCTTAA. Now, not miraculously, but it's a very nice thing to think about. You can see here that this overhanging strand from the foreign DNA is complementary to the overhanging strand from the vector DNA. Because you can see here, A is complementary to D, A is complementary to D, and these two Ds are complementary to these two A's. Therefore, if you have these two DNA pieces together, they can join by complementary base pairing, and hence you have successfully joined 2 pieces of DNA. Now, this ability to join 2 pieces of DNA becomes extremely important, as you will see. You'll be able to appreciate later. So, what do we use these restriction enzymes for? you use them to cut DNA at specific locations. So you can govern which locations are being cut by these restriction enzymes, which facilitates you to join together different DNA pieces, which is essentially remarkable. Before these enzymes were discovered, you did not have a way of joining two DNA pieces like this. So essentially, you need both sequences to have the same. You need both sequences, your vector and your foreign DNA, should both have the recognition sequence for this particular enzyme for this to work. You can easily appreciate this. And then this will produce an overhang here. So GCTTAA. Okay, this is called an overhang for obvious reasons. GCTTAA. And these join. These are also called sticky ends for the very reason that since they are both sticky, you can kind of glue them together. That also makes sense. Gluing here means joining by complementary base pairing. So you call these overhangs sticky ends. Of critical importance is that these will only be sticky ends if the same restriction enzyme cuts both DNA pieces. Because you must remember that each restriction enzyme has a specific recognition sequence. So EcoR1 produces this specific overhang, which would not be sticky to an overhang produced by another restriction enzyme, because that restriction enzyme would have a different recognition sequence. Therefore, it would make a different overhang when cutting. Therefore, these two ends would not be complementary to each other and they would not be sticky to each other. So you can see here, the enzyme cuts both DNA strands at the same site. That is a property of the enzyme itself. And this is essentially the definition summarized very nicely for this example. EcoR1 cuts the DNA between bases GNA only when the sequence GAA-TTC is present in the DNA. Therefore, if you have two different DNA pieces, you can generate sticky ends and you can join them together. So this is the general idea. And now we need to look at some nitty-gritties. So when you talk about restriction enzymes, these come under the broader class of enzymes called nucleases, which are called endonucleases, and the broader class of enzymes called nucleases, which can be divided into endonucleases and exonucleases. As you can see, endo means inside. So endonucleases make cuts at specific positions within the DNA. So they make cuts at specific positions within the DNA. Therefore, EcoR1 is an example of a restriction endonuclease, because wherever the sequence GAA-TTC, CTTAG is present inside the DNA, EcoR1 makes a cut at those sequence sites. So these are endonucleases. So EcoR1 is an example of a restriction endonuclease. It's a restriction enzyme and it's an endonuclease. So EcoR1 is an example of a restriction endonuclease. And the other type is exonucleases, which are not important right now, but suppose you have a stretch of DNA. Exonucleases can remove specific nucleotides from the ends of the DNA. So exo means more on the outside, so towards the ends, and exonucleases can remove nucleotides from the ends of the DNA. Whereas endonucleases, such as EcoR1 and many others, make cuts at specific positions within the DNA. Now, how do you name? This is important for your exam, because for your NEET exam, because they could ask questions based on nomenclature of restriction enzymes. The order is very simple. What is the organism from which it is isolated? Which strain of that organism? And what is the, let's say, suppose there were more than one enzyme isolated from that strain. So which, was it the first or the second or the third? That is essentially how you name a restriction endonuclease. For example, we just discussed EcoR1. Eco stands for E. coli. So you must know that this enzyme came from E. coli. That's the first bit of information. Next, okay, so which strain of E. coli did it come from? R indicates that EcoR1 came from the R strain of E. coli, and 1 indicates that it's the first restriction enzyme to be isolated from the R strain of E. coli. So now I'll try to provide a short clip next, which just maybe a very short 2 to 3 minute clip, which explains the basics of restriction enzymes in more detail, as in from where did we get them? So now you're wondering. So it came from E. coli. So it's safe to presume that these restriction enzymes came from bacteria. So what are they doing in bacteria? What is their importance in bacteria? So then once we discovered them in bacteria, it's quite genius that we were able to exploit them for our purpose of joining different DNA pieces together. So I provide a short clip where I explain a little bit more detail about these restriction endonucleases. But for the purposes of this lecture, you should know that the nomenclature is that the organism from which it was isolated, so in this case E. coli, which strain of that organism, so in this case the R strain of E. coli, and is the first enzyme to be isolated from the R strain of E. coli. And another very, very important point which I need to mention is a peculiar property about the restriction sites of these restriction endonucleases. It is to be noted that the restriction sites of restriction endonuclease, the recognition sites of restriction endonucleases are palindromic. So excuse me for the poor pen. I wish I could type it out more clearly, but they are palindromic. What does palindromic mean? So a palindrome is essentially something which is the same when read forwards or backwards. For example, ABA, when you read it forwards, ABA, when you read it backwards, also ABA. What this means is that we know that DNA runs anti-parallelly from the 5' to 3', from the 3' to 5' direction. So a palindromic sequence is one that essentially, if you invert the 5' to 3' side, 3' to 5', and you make it 5' to 3', it reads exactly the same as the 5' to 3'. And if you reverse the 5' to 3' and make it 3' to 5', it reads exactly the same as the 3' to 5', which is very clearly illustrated when it comes to ECOR1. So this is 5' to 3'. Let's flip the top. So let's flip GAATTC. What do you get? You get CTTAAG, which is essentially the bottom sequence. Flip the bottom strand, you get GAATTC, which is the top strand. So the recognition sequences of restriction enzymes are palindromic in nature. So to summarize what we need to know, this is an important restriction enzymes are, well, first of all, they're proteins. In layman terms, they can be called molecular scissors. They're an important tool of recombinant DNA technology. They essentially cut DNA at specific well-defined sites, where a site is a location on the DNA and it's defined by its length and its sequence. And then a particular restriction enzyme cuts DNA wherever this site is present along the entire stretch of the DNA. How do you name them? You start by the organism from which they were isolated. What is the strain? In this case, the R strain. And then what is the order in which the enzyme was isolated from that strain? And then they recognize these particular sites, which are called recognition sequences, or they recognize these particular sequences, which are called recognition sequences. And these are palindromic in nature, which means if you take the 5' to 3' strand, the sequence in the 5' to 3' orientation, and flip it, then you get the exact same sequence as the 3' to 5' orientation. If you flip the 3' to 5' orientation, you get the exact same sequence as the 5' to 3' orientation. And they can be used to join DNA pieces, disparate DNA pieces, as in different DNA pieces. As so, when you cut both DNA pieces with the same restriction enzyme, you produce the same kinds of sticky ends or the same kinds of overhangs, which can join by, let's say, complementarity mediated base pairing, and you can get recombinant DNA. Now, this is a kind of a higher level example. Please ignore most of the different terms used in this slide. I just wanted to highlight how this could be useful. Let's say, so this is the insert. YGOI stands for your gene of interest. Say that this gene is flanked by ECOR1 and NOT1 restriction sites, so specific sequences which can be recognized by these enzymes. So what you can do is you can take this, it's called a plasmid. Ignore what a plasmid is for now, we'll get to that. You take this plasmid and you digest it. Essentially, that means you introduce these enzymes at the optimal conditions for their functioning, that's known as a restriction and digestion. You digest it with these two particular enzymes, ECOR1 and NOT1, which creates cuts on either side because their recognition sequences are present here. And you get this free insert. And here, so this is your donor plasmid, this is your recipient plasmid, where you have the same sites, ECOR1 and NOT1. You digest this with ECOR1 and NOT1. And this creates overhangs in the recipient plasmid, which are complementary to those from your insert, because you digested both pieces of DNA with the same set of restriction enzymes. Cannot stress how important that is. If you were to use, let's say, HIND3 here instead of NOT1, then you would have one side which has a complementary sticky sequence to ECOR1, but this would be a HIND3 sequence, and in the recipient, and it would be NOT1 in the donor, so you wouldn't be able to join the DNA sequences. And then this process by which you join these pieces is called DNA ligation. So you ligate this insert into this recipient plasmid, and therefore you have successfully taken this DNA piece from one plasmid and introduced it into another plasmid. That's essentially what's happened. You've transferred this DNA piece from one plasmid to the other. Ignore all the other complicated things written in the slide. Just wanted to highlight that you can use more than one restriction enzyme in combination and use these to transfer a DNA piece from one specific sequence, or in this case plasmid, to another plasmid through a process where you start with digestion. It's called a restriction digestion because you're digesting these plasmids with your restriction enzyme, followed by ligation. Now, another important thing I want to highlight is restriction fragment ends are not always sticky. There are different kinds of ends. One is sticky, and they can also be blunt. So we already saw ECOR1 cuts between GNA. And yeah, so this is, you can see the 5' phosphates here. You can see the 5' phosphates, and of course the other side is the 3', so you have no edge, so you don't represent the phosphates. And you see this sticky sequence here. And you can also have enzymes such as EcoR5. Again, the name suggests this is the R strain of E. coli, and it's the 5th enzyme. So you had EcoR1, 2, 3, 4, and then 5. It's the 5th enzyme, restriction endonuclease isolated from the R strain of E. coli. And this has its own palindromic sequence. As you can see, GAT-ATC. You flip the top, you get CTA-TAG, which is exactly what the bottom is. You flip the bottom, you get GAT-ATC, which is exactly what the top is. This cuts it in a more blunt manner, which means wherever you get the site, it cuts it between this TAAT and you get blunt ends. So remember, restriction fragment ends do not always have to be sticky. They can be blunt as well. Okay, so this is kind of where I want to, this is the last slide, this again from your textbook, which is how you can ligate foreign DNA to a plasmid of interest. So what you see here is, you have your foreign DNA here. Now, one thing I want to emphasize is the book makes it look very simple. So you have this neat foreign DNA here, then you cut using the same restriction enzyme as I mentioned before, where you cut both the foreign DNA and your vector DNA. Again, I'll explain, come to what vectors are later. So when you cut it with the same restriction enzyme, you get this piece here, and you get this here, which have the same sticky ends because you cut it with the same restriction enzyme. Then you ligate it. So then you get this particular piece in your plasmid. This is now a recombinant DNA. It's a recombinant, again, excuse my handwriting please. This is a recombinant DNA because you've introduced a new piece of DNA. And then when you can introduce this into E. coli, and this plasmid can divide in E. coli. So therefore you can have this particular gene expressed to produce protein in E. coli. All is well and good. But then you should think, how do you get this foreign DNA perfectly like this? Is it really that easy? Is it that perfect? Can you get things that perfectly? Can you get a foreign DNA, in this nice configuration here? Is it really so simple? Just digest and take this particular piece out. What other considerations you have to take to ensure that the process is as smooth? So you should always keep in mind, whenever you see a figure like this, it looks very smooth and clean, but there are in fact it's a very thoughtful and considered process that goes behind, doing this actually in a research lab, which is something you'll appreciate if you move forward into research. But for the purpose of your examination, you can think of it as this very neat and clean process where you have this nice foreign DNA, you digest it with a particular restriction enzyme, take out a particular piece, digest your vector with the same restriction enzyme so that you get a nice slot here where you can ligate this particular piece into the vector, then use it to transform bacteria, and then the cells divide. Therefore, this piece can now divide in E. coli. So you are able to transfer this particular plasmid onto the progeny, and this plasmid has this particular DNA piece. Therefore, that piece is also transferred onto the progeny, and that's how you've achieved this recombinant DNA. You've joined one particular piece from foreign DNA into your vector. So with this, I'd like to conclude this lecture on the first tool, which is recombinant DNA technology. And in the next lecture, we'll start looking at the second tool of recombinant DNA technology, which is agarose gel electrophoresis. Thank you for listening, and bye.